lockdown, so I'm joining you today from my kitchen, a few miles north of here. So I'm going to talk about signaling through acidic calcium stores. So in the last talk, Don Gill was the guinea pig. He told us about calcium entry, so we're changing focus now to release of calcium from intracellular compartments, in particular acidic compartments. So acidic calcium stores, in terms of background, they're a collection of organelles found in animals and plants and elsewhere. They're all different morphologically in terms of size and canonical functions, but we can group them together under this umbrella term, acidic calcium stores, due to their proton-rich and calcium-rich interior. I'm going to talk predominantly about calcium signaling through lysosomes. So lysosomes, highly acidic organelles, pH of around about 4.5. Their calcium concentration, 500 micromolar, pretty similar to the free calcium concentration in the endoplasmic reticulum calcium stores. And we know about lysosomes and their canonical functions as degradative centra. It's clear now that they have many other functions, and calcium signaling is one. So we've been studying extensively the two-pore channels, the TPCs, for many years now. So TPCs reside on the membranes of acidic calcium stores, and when activated, for example, by NADP here, they can release calcium into the cytosol to regulate cell function. So historically, it's about 10 years ago then, when three groups, including ours, converged on these TPCs as the likely target channels for the calcium mobilizing messenger NADP. So NADP has been known for many years to release calcium from acidic organelles. Initial work in sea urchin eggs from Anthony Gaglioni's lab, and then this has been confirmed subsequently in numerous other cell types. So one of the features over the last few years that's become clear for signaling through this pathway is that NADP releases calcium in a highly localized manner. And during signaling, these local calcium signals can be amplified into global ones, but it's clear now also that the local signals themselves are functionally relevant. And that functional relevance is exemplified if we consider what the endolysosomal system actually is. So we have inputs coming into the endocytic system through endocytosis. We have outputs through exocytosis. We have lots of trafficking events of vesicular fusion and fission. And we've known for a long time that these various events are calcium dependent, in fact, dependent on local calcium signals. So having calcium permeable channel like the two-pore channel on the membrane of these acidic organelles then provides us with a mechanism then regulating these various vesicular fusion and fission events and the associated cellular processes. And in fact, in this review, it's a bit out of date now, but pretty much all of these events that have been highlighted here on this slide have been shown to require TPCs. Just very briefly, some of our own contributions to this literature. One of the things that's become clear is that if you simply overexpress one of the two human isoforms of these channels, TPC2, this is some work we did with Jonathan Marchand a few years ago. One of the findings that other labs have seen as well is that the organelles expand. They look a lot bigger. So if you compare that to the organelles, if you were to overexpress a poor mutant, so we've shown that this poor mutant is electrophysiologically dead, then you don't see this expansion. And this expansion has been contextualized in the context of this fusion event. The proteomics of TPCs are consistent with fusion and vesicular trafficking more generally. And we think that this expansion of the organelles may be pathophysiologically relevant. So in some work here, again, a few years ago in a model of Parkinson's disease, this is a familial form of Parkinson's disease. It's become clear now over the last 5 years that many endocytic dysfunctions associated with the disease, including expansion and clustering of lysosomes. And what we showed in this paper was that we can correct this dysfunction by knocking down TPC2. This is isoform selective as well, which is quite interesting. Moving away from vesicular trafficking, it's also clear that TPCs play roles in non-vesicular trafficking. So for many years, this amplification that I mentioned, these local signals being amplified by ER calcium stores, is thought to occur at regions of close apposition between lysosomes and the ER. And here's some electron microscopy showing membrane contact sites between the two organelles, these little red areas highlighted here. The ER forms junctions with many other organelles, including late endosomes. What we showed again relatively recently was that the other isoform present in humans, TPC1, can actually localize that to the contact between late endosomes and the ER. And when we interfere with signaling through NAADP using this antagonist here, NED19, or if we knock down TPC1 but not TPC2, we can disrupt these junctions. And this has knock-on effects on global endolysosomal morphology and on growth factor receptor signaling, which happens at this locus here. So that's just a little taster on the function of TPCs within the endocytic system. There's many other examples out there. But one of the, and I've kind of, I guess I've talked about that in the context of these local calcium signals and their activation by NAADP. But one of the confusing things in the literature has been what the actual ion selectivity of the channel is and what the actual activating stimulus is. So those of you who've been following the literature will know that there is some very strong biophysical evidence suggesting that 2-pore channels are not permeable to calcium, but rather highly sodium selective, and that they're not activated by NAADP, but rather by phosphoinositide PI35P2. And this is a phosphoinositide which is enriched on the membrane of endocytic organelles and which we know is also important in membrane travel. So it was my view, still is, that these channels are co-regulated by NAADP and PI35P2. There's some evidence, I don't have time to go into this, that NAADP is likely to activate the channel indirectly through some sort of associated binding protein or receptor. So for me, it's not worrying that if some people don't see activation by NAADP, it could simply be due to loss of that factor. But what's much more confusing is the ion permeability or the ion selectivity of the channel. So lots of evidence, lots of independent studies suggesting that TPCs are non-selective cation channels which are permeable to calcium, but equally there's independent evidence suggesting that they're highly sodium selective. So there's something clearly up, if you like, with the ion selectivity of TPCs. And so what I'm going to do today is tell you about some recent published work that we did in collaboration with Christian Grimm, which supports this co-regulation model of TPCs by both of these ligands and provides some insight into the actual ion selectivity of TPCs. So there's a nice low-res picture of Christian. I think he's here. He's based at the LMU in Germany. And the work I'm going to talk about predominantly, the data that I'm going to present, is some work done by Cara in my lab. She's a final year PhD student who's in the final stages of writing up her thesis. So what Christian did was to generate a stable cell line expressing TPC2 at the plasma membrane. So this was based on an early observation from our lab that identified an endolysosomal targeting sequence within the N-terminus of TPC2, such that when you mutate that sequence, the TPC is rerouted essentially through the default pathway to the plasma membrane. And what he did was to use a calcium-based assay, high-throughput assay, to try and identify compounds which could mediate activation of the 2-pore channel. Because one of the real problems with working with these channels is their intracellular location and really trying to get access to that intracellular location. And possibly by breaking open cells, we're running into some confusion. So having cell-permeable activators of these channels would really help, and that was what Christian's aim was. So here's the results of his primary screen. Some of the compounds here. What you're looking at here on the y-axis is the response in the cells expressing this mutant TPC relative to a control cell. So if that number is bigger than 1, then you've got a selective response in that cell. And he identified 2 hits here, which he named TPC2A1N and TPC2A1P. And by the end of the talk, hopefully they'll become clear why they're called that. So the first thing Cara did then was to try and validate this finding. These are transiently expressed HeLa cells expressing that same poor mutant the plasma membrane version of TPC rerouted to the plasma membrane. She measured calcium globally here with FURA2, and what she found was that when you stimulate the cells with TPC2A1N, you can indeed get a nice calcium signal in these cells. She took away extracellular calcium from the media, repeated the experiments, and as you can see here, there is now no response to the compound, suggesting that the compound is mediating calcium influx across the plasma membrane. I should say that by rerouting the channel to the plasma membrane, you maintain the topology of the channel such that the cytosolic bit of the channel remains cytosol. What Cara also did was use a poor mutant. Mentioned that previously in terms of the morphology changes that we can see upon overexpression of TPC2. Here she's coupled the mutation with this endolysosomal targeting mutation. So we've got a channel dead version of TPC2 at the plasma membrane. And as you can see on the right-hand side here, there is no calcium signal in these cells. And finally, what Cara did was to do similar experiments with cells expressing tripmucolipin-1, another lysosomal calcium channel, again tricked to the plasma membrane by interfering with its targeting motifs. And what you can see here is that A1N again has no effect in these cells, but you can still activate calcium signals by its exogenous activator. So all in all then, that suggests that this compound is selectively evoking pore-dependent calcium influx through TPC2. Moving on to some pharmacology. This was a very nice paper published in 2015, quite topical now in terms of viruses. What this paper showed was that the Ebola virus required TPCs for its entry into cells, such that when you block TPCs, either molecularly or, as I'm going to go on to, chemically, you can essentially trap the Ebola virus in the endocytic organelle, this little worm structure here, and prevent infection. And this compound emerged from that paper, tetrandrine, better known as a calcium channel blocker, a voltage-gated calcium channel blocker previously, but in this paper they showed that tetrandrine was particularly potent at blocking the TPCs. And generally we'd known the year before, and actually for some historical data back in the sea urchin homogenates, that TPCs or NADP action could be blocked by voltage-gated calcium channel blockers. So this is kind of fitting with this idea that is a loose pharmacological profile, if you like, for the TPCs. So here are some experiments with that compound. Here's the control, TPC-A1N, this compound causing a nice calcium signal in the cells, as we've seen previously, but a relatively low concentration of tetrandrine added acutely can block that signal. Following the publication of this paper here, or actually I should say during this outbreak, the first of the outbreaks of the Ebola virus, there are many papers like this which appeared in the literature. You see this now with COVID as well, where efforts were made at trying to repurpose existing drugs to try and block viral entry. So at that time, we were also doing some screens to try and find blockers of TPCs. We were using a virtual approach by modeling the pore of TPCs here and then screening approved drugs in silico to try and find drugs which would dock into the pore and therefore could be used as potential pore blockers. And what we reasoned was that these wet screens here, these real screens, if you like, would yield drugs that potentially could be blocking Ebola virus entry through blocking the TPCs. So we cross-referenced our screen with these screens here to identify a set of common compounds. And reassuringly for us, or making life a little bit easier, these compounds tended to group into being either dopamine receptor antagonists or selective estrogen receptor modulators. And then we went on and showed in this paper, I don't have time to go into this, that these were indeed blocking channel activity. So Cara tested these compounds in this assay here. This is one of the dopamine receptor antagonists, and as you can see here, it blocks the calcium signal in response to that compound. She also used raloxifene, a SERM, a selective estrogen receptor modulator, and again, that calcium signal was blocked, providing further pharmacological evidence then that this calcium signal is through the 2-pore channel port. And just very briefly, in terms of selectivity, here's some pool data with the TPC2 in the plasma membrane. Here are the control responses in black, and in red are the inhibited signals in response to the 3 drugs. And what's nice here is that we can do parallel experiments with triple ML to probe for specificity. And again, as you can see here on the right-hand side, none of these drugs are blocking the effects of activation of that channel. Okay, so as I say, there were 2 hits that Christian found in the screen. So has just died. Here we go. So we also did experiments with the second compound, A1P, and this is where things got even more interesting. So like the first compound, the N compound, the P compound also causes a calcium signal in cells expressing TPC in the plasma membrane. It's due to calcium influx because it disappears when we take away extracellular calcium. It's due to pore activity because we don't see it with the pore mutant. And it's selective because we don't see it in cells expressing triple ML. But one difference between this compound and the other compound was this big delay here. So response to the N compound wasn't particularly quick under these conditions, but the P compound was dramatically slow. And you can see that here. This is a maximal concentration of the agonist. The EC50s are about the same, and so too are the maximal responses. And you can really appreciate this kinetic difference between the N compound in black and the P compound in red. So it appeared then that the P compound certainly was activating the channel, but it was doing so in a weaker fashion. So thus far, I've been telling you about the effects of Christian's compounds in the plasma membrane. But of course, TPCs are normally located within the cytic system. So we had a look to see if these compounds would activate the channels in the endolysosomes. So using the construct here, TPC2 fused to GCAMP6S, genetically encoded calcium indicator. This is on the C terminus here. And in cells transiently expressing this, where it gets some nice punctate staining, N compound causes a nice calcium signal in these cells, telling us then that the compound must be cell permeable, and again, supporting this idea of calcium permeability. And we can couple that pore mutation, the L265P mutation, into this fusion, and we lose that calcium signal that's shown here in red. Similar experiments with the P compound here. Again, we see a signal. It's a lot smaller. So once more, the P compound doesn't seem to be as good, if you like, as the N compound. But what we do see is pore dependent because it's lost in pore mutant. And once more, we can check for specificity here. Do similar experiments with triplicylipin 1 fused to GCAMP, shown here in the bottom, and neither the N nor the P compound were able to cause a calcium signal in these cells. But again, exogenous activating the positive control would activate them. Okay, so all in all then, I think these data support the idea then that TPCs are calcium permeable. They're in intact live cells. Nevertheless, these are indirect measures of channel activity. Of course, we need to see what these compounds are doing at the channel level. And this is where Christian's expertise really shines through. He's one of the few labs in the world that can record electrophysiologically from lysosomes within cells. Now, strictly speaking, they're not really lysosomes. They're enlarged structures which can enlarge them chemically in order to apply the patch clamp technique. And on the right here, these are Christian's electrophysiology experiments. And what he found was that both the N and the P compound can indeed induce currents in these large vacuoles expressing the wild-type channel. So confirming that these channels are indeed activated by the agonists. But what was really interesting here was that the P compound was inducing larger currents than the N compound. So this is kind of opposite to what we were seeing with the compounds in the live cells. It's kind of quantified here. These are two different variants of TPCs. On the left here is the responses to N compound, and on the right is the responses to the P. Notice the scale difference here. Now, these electrophysiological recordings were done using sodium as a permeant ion, whereas in the cells, we were using calcium as a surrogate, if you like. So Christian reasoned that perhaps these two compounds are inducing different sodium and calcium permeability. So we had a look at that in the live cell. So what Cara did was this experiment here, where she went back to the cells expressing TPC in the plasma membrane. She measured calcium in response to the N and the P compound, but she compared the responses by removing extracellular sodium from the media, replacing it with NMDG. So for the N compound, the calcium signals are not much affected. They do get quicker when you remove sodium. What was quite striking was with the P compound, this delay that we were seeing before is completely gone, and the calcium signals are much larger in the absence of sodium. So one way of interpreting that data is that perhaps the ions are competing for the pore, permeable to both sodium and calcium. Now, when you remove sodium, you promote calcium flux, and that this is more pronounced for the P compound. So to test then more directly whether there are actually, if you like, changes in the ion selectivity, Christian did this key experiment, which is to look at the relative permeability to sodium and calcium in these vacuolar patch clamp experiments. These are bi-ion experiments, calcium in the lumen, sodium on the outside. So under these conditions, again, both of the compounds can induce currents, which is great. But notice here, the reversal potentials are completely different for the two compounds. See that a little bit more clearly here if we zoom in on these traces. So the P compound is definitely negatively shifted relative to the N compound. But this is telling us then that these agonists are behaving really quite differently when it comes to the relative sodium and calcium permeability. This essentially looks like two different channels. So another thing that Christian did here was then to compare this to the activators of the channel, the endogenous, the physiological, and proposed activators of the channel. So here are the data with NADP here on the left and with PI3-5P2 on the right. And what you can see here is that, again, both of them could give a current. PI3-5P2 current is something that's reproducible in many labs, but as I mentioned, the NADP induced stimulation of the channel comes and goes. But certainly under these conditions, it's there. And what you can see, hopefully, is that the characteristics of this curve are very similar to the N compound. See that there. Whereas the P compound seems to be mimicking PI3-5P2. You can see that. So what is this telling us? Well, this is telling us that, as I say, that the sodium and the calcium permeability must be different for the two different agonists, whether they're exogenous or endogenous. So we can convert these reversal potentials into permeability ratios, shown here in this slide. So this is the permeability to calcium relative to sodium. And with the N compound, or NADP, hence the name, that value is close to 1, i.e. TPC is behaving as a non-selective cation channel. In contrast with the P compound and PI3-5P2, that ratio is much, much, much lower. This is essentially telling you that the permeability to sodium is much higher, i.e. that it's behaving as a sodium selective channel. So it's the same channel, it's TPC2, yet it seems to be behaving completely differently depending on how you activate it. So going back to the intact cells, then we thought, well, can we try and get something that mirrors this in a live cell setting, if you like. So back to the influx experiments, this Ca2. What Ca2 did here was to measure calcium here on the left, but also measure sodium on the right. So with calcium, as we've seen before, this is the same concentration of agonist this time. So amplitude is a little bit lower for P, but that delay is there. But when you measure sodium in the same cells, you can see here that now there's no longer a delay and that the amplitude of the sodium signals are very similar. I think what this is kind of telling us, this is poor man's electrophysiology, if you like. The sodium signals are similar, but the calcium signals are weaker for the P compound. In other words, the P compound is more sodium selective, if you like, than the N compound. Kind of corroborates the findings with the electrophysiology. So going back to this model then, I think this is not so far off, that the channel is indeed co-regulated by the ligands. But what we have now then are small molecules, cell-permeable molecules, which appear to be mimicking the actions of NADP or PI3-5P2. So with NADP and the N compound, the channel seems to be non-selective, letting in lots of things, whereas in the presence of the P compound or PI3-5P2, there seems to be a clear sodium bias. And we can see that both in intact cells and at the channel level. So overall, I don't think we're far off from pretty much what was published previously. So nobody was wrong. So when the channel is activated by NADP, you get something that's biased towards calcium signaling, but when you activate it with PI3-5P2, it's biased towards sodium signaling. Okay, so I think I've probably still got a couple of minutes. Just want to talk a little bit about the mechanism of the action of these compounds. As I mentioned, NADP seems to work through some sort of associated binding protein. Now, that binding protein has not been identified molecularly, so it's difficult to get at that using a molecular approach. In contrast, PI3-5P2, its actions have been characterized molecularly, in fact, structurally. My slide responds. We have structural information on PI3-5P2. It binds directly to TPC1, as shown here, and also to TPC2 about there, which in this format is here. This is between the S4 and S5 linker. So in this experiment, what Ca2 did was to mutate one of these residues within this binding pocket, Ca2, and then compare the calcium signals, calcium influx in this case, in response to the N compound. So as you can see on the left here, the N compound responses are still intact in the presence of that mutation. But what was quite striking was that the effects of the P compound are definitely reduced. So it's the same mutation responding differentially to these two compounds, suggesting that the P compound is interacting at a site similar to PI3-5P2. There's some quantification there, lack of effect of N, and a reduction with P. She also did this at the lysosomal level with the GCAM6S construct. Again, stick the mutation here, A204. On the left, the responses to the N compound are no different, whereas the P compound, the responses are modest to start with, but then pretty much reduced here in this particular example upon mutation. And that's quantified here. Significant difference for the P compound, nothing for the N. And finally, back to the electrophysiology in Christian's lab. Again, here are the currents. The N currents are not affected by mutation of Ca2, whereas the effects of the P compound are significantly reduced. So, in summary then, new cell-permeable TPC2 activators have been discovered, which appear to mimic intracellular physiological cues in switching the ion selectivity of TPC2. That's the sort of take-home message, if you like. What I've shown you is data showing that the N compound is an NAADP-like calcium agonist, for want of a better term, whereas this second structurally distinct P compound is more like PI35P2 and is sodium agonist. And I've shown you a little bit of a mechanistic, giving you a bit of mechanistic information showing that the N and the P compounds appear to target distinct sites. So implications then, I think this resolves conflict on the permeability and gating of TBC2. Nothing to fight about now. Also, physiologically, what this means is that we have an example of a single ion channel which can mediate very distinct ionic signatures depending on how the channel is activated. So that, we suspect, will have major physiological consequences for how the endolysosome regulates cell function. So the paper is published now. Finally, this was a hard slog to get this out, but it's out now. Lots of people involved in this work. As I say, this is a study led by Christian Grimm. I just want to highlight all the joint first authors here. So I've mentioned Cara already, but Suzanne from Christian's lab, who's the chemist, Maxo, Cheng Chang, and Yu Kai with the electrophysiologists. So there's another picture of Cara again, looking happy. She's not looking so happy now because she's writing up a thesis, but she'll be happy soon. Christian, again, and other collaborators. Thank you all for listening.
Thanks very much, Sandip. Really, really clear. Number of questions have been pinging away already, so let me just work my way through the ones that perhaps you haven't answered. Actually, a lot of these have been already asked. A few questions, one from Anthony Gaglioni, obviously, asking about proton conductances. Do protons go through TPCs?
It would appear so, yeah. So where are we? Yeah, so Christian did experiments with protons to see if it's proton permeable. There is a small current, you can see here on the left, in response to the N compound, where you don't see it with the P compound. So this would support the idea that in the presence of the N compound, the channel is acting as a non-selective cation channel. Okay, thanks. And functionally, I think also, if you go and look at this paper, there's some very nice experiments in there done by a collaborator, Christians, where they measured pH changes in the endolysosomal lumen, and the N compound does seem to change the pH of the lumen, which might be consistent with this proton permeability. And again, it's agonist selective.
Okay, thanks. A question from Peter Stathopoulos in Western University asked the question, how similar are the compounds structurally to each other and to their respective NAADP or PIP2?
They're all different. So the compounds are structurally dissimilar to each other, and they do not look like the cognate compounds either.
That's in 2D, but I mean, as we all know from the original NED19 compound, the three-dimensional shape and charge can look very different in 2D. Have you checked to see if the model in 3 dimensions is closer?
Not extensively, but again, they do not look similar to our eyes.
Oh, okay. Okay, that's fine. A question from Christoph Brenker. Have you put N and P compounds together? Have you added them simultaneously?
Yeah. So that's experiments on the go at the moment. It's not published yet, but just say one thing. So in Christian's electrophysiology experiments, so going back to these reversal potentials, what's shown here is the change in the reversal potential when a PI35P2 is added after the agonists. So in this example here, this is the reversal potential after first the channel is stimulated with the N compound, and then PI3-5P2 is added. So in essence, you kind of start here, then you add the PI3-5P2, and you can see there's a negative shift in the reversal potential. So it's going towards that. And that's true when added after the N compound or NAADP. Whereas you can go the other way if you start off with the P compound. So the reversible potential can toggle in the presence of the two, but we're looking more carefully at exactly what the extent of that toggling is and if there's a dominant compound, etc.
Okay, thanks. A question by Rajesh Bardwaj. When TPC2 is at the cell surface, if there's no 3,5 in the plasma membrane, why does the P and N compound show difference in that assay with the Ca2 mutant?
So yeah, that's a good question. So yeah, absolutely. So in the plasma membrane, obviously it's not in its native environment. So we wouldn't expect huge amounts of PI3-5P2. I think what those data suggest is that both compounds can act independently of PI3-5P2 in the first instance. In other words, you don't need the PI3-5P2 there to get channel activity. And secondly, the mutation makes sense if the P compound were interacting at the same site as PI3-5P2 would normally activate the channel. So I don't see a discrepancy.
No, I agree on that one. In the absence of Anthony has asked, structure activity relationships of the compound seems all or nothing. Is that correct? I don't know what that means.
So I think, is that Galeoni, Anthony Galeoni? Yeah. So I think he's probably referring to the published paper where there are a whole series of analogs of the different compounds. And yes, I think it is a bit all or none at the moment. We haven't managed to increase the potency of any one of the compounds. So there's some interesting bits where we can show which bits of the compounds are required for activity. But yeah, there's nothing more subtle coming out of that at this point.
Okay. Guy Ruash has asked a nice question. Do you think the N compound mediates the activation of TPC, requires an accessory protein similarly to NAADP, or is it a different mechanism of activation that converges on that?
Yeah, that's a very good question. So that's something that we're trying to work out now, exactly what's going on. One piece of data I didn't show you was that these compounds seem to be selective for TPC2 over TPC1. So that might suggest that the compounds are working directly on the channel because both TPC1 and TPC2 are reportedly activated by NAADP. So that's an indirect piece of evidence to suggest that the N compound might be acting directly on TPC2. But at the moment, we don't have any further data. It's something that's in progress at the moment to try and find where the binding site is.
Yeah, of course. I'm not going to press you on that because I imagine that's a pretty hot topic. Can I ask a sort of a more general question? In terms of the differences in the ion selectivity differences clearly between the N and the P, or between PIP2 and NAADP, one is presuming that you're going to get a different modality of signaling, one through sodium and one through calcium. How do you envisage sodium-dependent signaling working in a lysosomal system, in a vague sense? I don't mean in a specific sense.
Yeah, I think thinking about that a lot. So when the original PI3-5P2 regulation of the channel was identified by Haoxing Su and Daejeon Ren, they put forward the idea that perhaps sodium flux through the TPCs could set the membrane potential. So maybe that mode of signaling is through setting the membrane potential, which would have many knock-on effects on, for example, acidification, the circuits required for counter fluxes, for fluxes through other ion channels, chloride channels, as an example, and perhaps other cation channels. As you know, the list of ion channels on the lysosome are increasing. So one idea might be that it sets the membrane potential, which in turn then has a secondary effect on other ion transport mechanisms.
Yeah, sure. Thanks. Do you think there are, I can't think of many other precedents in biology where a ligand is changing the ion channel selectivity? I know that there's some protein-protein interactions, but a small ligand, is this unprecedented?
Again, we tried to find examples of ligand-gated ion channels which switch their selectivity, and we couldn't find anything. So if anyone knows, please email us and let us know.
Yeah, true. I mean,
as you say, the protein-protein interaction thing. So, you know, STIM and ORI, for example, the stoichiometry of STIM and STIM to ORI appears to affect its ion selectivity. Again, the mitochondrial uniporter, I think there's some evidence suggesting that BQ1 can regulate manganese permeability. Not in terms of physiological ligands, but again, going back to store-operated channels, 2-APB has all sorts of interesting effects on ion selectivity there as well. Yeah. P2X receptors, they're ligand-gated. They seem to change their ion selectivity, trip channels, but not different physiological ligands. We're not clear on that.
I don't know whether if you or if Christian is, I know, online today. What about TPC1 with NAADP and PIP2? Does the same thing happen with that? I'm not quite sure, because that I certainly know that it's sodium selective with PIP2. So I don't know whether the ion selectivity has been checked for that. Can you remember
No, that's a good question. We don't know if TPC1 is able to switch its ion selectivity. However, there's work from Rebecca Sitzer-Payson, who, using bilayers, presented some evidence that the PCA to NA was changing modestly in the presence of PIP2. So, but under those conditions, PIP2 was not directly gating the channel. It seemed to be more of a modulator. So maybe there's something missing in that.
Yeah, because I was obviously getting at the idea of if the N compound is selected for TPC2 and it seems to be acting at a different site, at least circumstantially from TPC1, because there is the selectivity, then I was just sort of trying to retrofit it to see if the same thing would happen with the TPC1. I mean, it then begs the question: are other lysosomal, is mucolipin 1, which has some structural homology, going to be, subject to a similar switch? Although we don't know anything other than PIP2, at least to the best of my knowledge, that's endogenously activating it.
Exactly. So but possibly with the exogenous agonist, there may be some differences, but relating that back to physiological activators, I think, is a key question.
Yeah. Okay. We have another question. I think you already answered it in the talk. This is from Rajesh again. He said, are the P and N compounds specific for TPC2 or 1? And I think you said that they were definitely specific for TPC2. Yeah, no, that's fine. You'd apologize. Yeah, you missed it. That's fine. No worries. It's easy. Because you didn't actually show the data.
No, I didn't show that. No.
You didn't show the data, so it's easily missed. No problem at all. Okay, Rebecca Burton has asked, under physiological conditions, what is the cue for TPC2 to allow sodium-calcium selective permeability, so for sodium over calcium? In other words, I think she's asking, what is the cell stimulus that would promote TPC2 sodium selectivity over calcium selectivity? What couples to it? I interpret that.
So, sorry, say that again?
Sorry, I think the question is, which cell stimuli couple to which different mode of the channel?
I see. Yeah.
Sorry.
Yeah, I guess. I mean, there's quite a lot of literature suggesting that physiological stimulation of cells with hormones and neurotransmitters can increase cellular NAADP levels. So one would imagine that they would bias it towards the channel behaving as a non-selective left-hand channel. There's less evidence about PI3-5P2 levels changing, at least acutely. There is some, but not huge amounts. Yeah. So you can imagine a scenario there where maybe that would bias it towards PI3-5P2. But I think to really answer that question properly, we need to know what the relative levels of PI3-5P2 are on the channel relative to NAADP. How quick those kinetic changes are, which I think they are pretty quick, so they can toggle for sure. But that's going to be a hard one to get at directly, I think. I think one approach to at least get at the physiology would be to look at channels which are deficient in PI3-5P2 signaling, for example, by knocking mutations and see what's left, if you like.
Yeah, exactly. I mean, it's a shame that the promised sort of PIP, the 3-5 sensing domain has been subsequently shown to be less selective than anticipated. So as a sort of a pH domain type, fluorescent pH domain type of approach in it, and Tamas Bala showed that that didn't actually, unfortunately wasn't as good as initially hoped, which just shows you that the selectivity of these binding sites for different head groups of the inositol lipids they're exquisitely selective, and therefore it's hard to make probes, because ultimately that's what you want to do, is you want to have a look at, be able to monitor the probes at the surface. And indeed then that may not still tell you what the relevant concentration is, if it's actually sequestered or etc. etc. I mean, Anthony Gaglione has made the point is that PIP2 seems to modulate a lot of lysosomal channels, whereas NAADP is unique to TPCs, which is...
I think this is something we've discussed a lot. I think my feeling is it's a cofactor. I still don't think that it gates the channel in its traditional sense. It certainly does in an electrophysiological context. It behaves precisely as you would expect as the ligand-gated ion channel, but I just can't see how that would work in a physiological setting, because you'd have to have lipid exchange coming in and out between the channel and back out again. I don't know how quick that is. So I still feel it's a more of a sort of slower modulated setting, the gain and or the ion selectivity. But I don't have any direct evidence for that.
No. And I think it's as you say, it's experimentally it's almost not intractable, but it's pretty challenging to get these things. Oh, have we just gone? I think my computer has just hung up.
I can still hear you. We can still hear you, Anthony.
You still hear me? Oh, right. Yeah. No one is moving. You're all frozen in time. So, okay.
You're still moving.
Yes. All right. That's fine. Still here. So Christian himself has asked a question. I don't know whether we want to keep going with questions or whether we want to now break it out into a free-for-all sort of coffee time session, or do you think that's too early, Martin?
I think what I'll do is I'll stop the recording at this point, and we can, if Sandip's prepared to just hang on and chat, then people can stay and indeed activate their microphones, because we've got a little bit more time now, and we can chat that way. I hope that's okay with everybody.
Yeah, that's fine. Thanks very much, Sandip. Really interesting.